The first speaker of this afternoon is Peter Mills, who is already in place. So Peter Mills is the Assistant Director at the Nuffield Council on Bioethics, an independent body which aims at examining and reporting on ethical issues in biology and medicine. Peter also used to sit on the Council of Europe Bioethics Committee as the UK delegate. Also in the past, he was head of human genetics and bioethics at the UK Department of Health. And Peter is going now to have the difficult task to address some of the main human issues. You have the floor.
Paul raised the question of what the role of ARRIGE could be in a debate that's been going on, as Hervé said earlier, since at least 2015, when CRISPR-Cas9 genome editing systems really caught the public imagination. I'd like to try to broaden out the discussion a little bit from what we've said what's been said this morning, and in order to do that, I just really want to make a few observations that have struck me about the debate and the way it's developed over the course of the last few years. So I've got 6 slides, and I'll try to rattle through them as quickly as I can. First question I wanted to ask is, why is genome editing of particular interest from the point of view of bioethics? There's a kind of conceptual question here, and the question is really, is it just a new tool for doing the sorts of things that we've been trying to do or hoping to do for a long time, and therefore raises issues that have been discussed, albeit in respect of other techniques, for a long time? Or does it raise substantially new issues? There are certain features of genome editing, recent genome editing techniques, that are novel. It is allegedly flexible. It is allegedly more effective than previous techniques, relatively rapid, relatively accessible to those with a good grounding in microbiology, relatively cheap, and as Francisco's slides showed this morning in terms of the publications, it is diffusing very rapidly within the context of research. So it's a powerful research technique. But what the question that I want to address is, under what conditions does that technique come to participate in what we might think of as new and emerging genome editing technologies? In a report that we wrote at the Nuffield Council a few years ago and published at the end of 2012, we defined biotechnology as an assemblage of knowledges, practices, products, and applications. And one of the observations that I would make about genome editing at present is that it depends, as do other technologies, on a great number of other enabling conditions, indeed enabling technologies, such as advances in genomic science. If it's going to be used within a clinical setting, for example, we need to know a lot more about potential genome targets. And in that respect, I think there are several kinds of challenges. There are technological challenges to do with genome sequencing, for example. There are data challenges in terms of whether or not it is even possible, if we were to sequence the whole, all of the people that are alive on the Earth today, whether we would have sufficient data to be able to focus in on some genetic changes of interest. And then there's the utility question, the question of whether or not any change, that clinical or embryological change that we make using a genome editing technique, how that's going to play out in the context of a living, functioning organism. So I think at this stage, there's a lot we don't know. We don't know whether we're going potentially to be able to do a lot with genome editing or just a very little. There are also questions of delivery, how you deliver the mechanism to the organism. Vectors, in the case of somatic genome editing, ICSI techniques, for example, in embryology. And even having considered all those technical questions, there are questions of the economic viability, regulatory approval, and of public support for genome editing, all of which I think are necessary in order for this to develop into technologies. So, when genome editing crossed into the public imagination, public consciousness, the immediate response from scientific elites was immediately to reject any notion that it was going to segue simply into clinical use. And what happened was a sort of process of segregation, what you might call segregation and linearization. And here there's just sort of three examples of that early response. The Don't Edit the Human Genome paper, which the idea of which effectively was to keep potential somatic treatments away from the germline prospect. The Baltimore paper, which was adverted to earlier today, and the statement from a number of UK research funders orchestrated by the Wellcome Trust, which essentially said, let's separate out the questions of research from the questions of what happens further down the line, and let's worry about getting an effective technique. Now, on the left, I've suggested that this segregation is undermined because there are a number of liminal spaces around genome editing. The first one is this notion of basic and applied research, and in some contexts, and I'm thinking particularly of the embryological context here, the notion that there is a distinct kind of practice that is involved in research compared to potential treatment. It may well be a distinction without a difference. In other words, the very same alteration or modification that one might make to a particular gene in a human embryo in a research context as proof of concept could immediately, were that embryo to be transferred, be a source of treatment. We came across this in relation to human cloning and the sort of essentially linguistic distinction between reproductive cloning and therapeutic cloning, which underpinned a lot of the early discussions there. There was very little interest in someone cloning a human being. There is much more interest in somebody altering a human embryo, and I think that's one of the questions that we want to think about. Scientists and there are others, the accessibility of human genome editing techniques, particularly CRISPR-Cas9, makes it accessible to potential sort of mavericks and competitors, people who want to get there first. There was a really interesting quote in the New York Times a while ago about where a researcher was saying it's really difficult to weigh up whether we should try to protect the interests of patients or get there first with our treatments. It doesn't seem to me that that's a very hard thing to weigh up, but the fact that it was posed in those terms, I think, indicated something about the anxieties that this generates. The global portability of technologies, of skills, of tissues, and of patients means that genome editing is not really potentially controlled by states anymore, and different forms of community may be involved in how it gets transferred. And finally, the innovation of genome editing techniques as technologies is now substantially invested with others' interests, with the interests of patients, with the interests, indeed, of the security community. I was involved in preparing a meeting that happened in Germany recently on assessing the security implications of genome editing technology. It's generated interest within the policy community in terms of laying down pathways for particular routes into treatment. And indeed, as somebody mentioned earlier this morning, even the DIY community, the DIY bio community, we saw Josiah Zayner injecting himself with a modified preparation only recently, and regretting it. So, that being the case, I would like to suggest that genome editing doesn't belong to, and nor is it controlled by, what one might think of as the Republic of Science in Michael Polanyi's terms. So the questions that we need to address ourselves to are not the ones that are focused on getting a safe and effective technology, but rather we need to ask questions about what we do with it when we have it. So not about the limited applications, the first applications, but about the possibilities. And the best way of asking that kind of question, it seems to me, is what will a world with genome editing in it look like? I think I'm going to skip over this quite quickly just to say that it seems to me that genome editing raises questions of three kinds when one comes to ethics. There are traditional questions of medical ethics involving individual rights, consent, those kind of questions. There are questions of public ethics and social norms. One often encounters anxieties about slippery slopes leading to unconscionable outcomes as soon as genome editing is proposed. And there are questions of global ethics, questions about whether or not certain uses of genome editing technology might undermine the nature of human being or infringe in important ways this notion that's very often mentioned of human dignity. And allegedly, and I'll talk about this a little bit for a few minutes, the issue of making heritable genetic changes, so-called germline interventions, is said to be one of these categorical limits. So, do we at least agree that there is a consensus on this third level? Well, here are three international instruments that have legal force of different kinds that are potentially relevant. The first one is the UNESCO Declaration on the Human Genome and Human Rights, and you'll notice that what that has in its sights is some notion of the structural integrity of the human genome as something shared amongst people. Of course, there's no living human who has this human genome. The human genome, if you like, is a constantly evolving, constantly changing. And the notion that, as has been mentioned earlier, that we might be infringing the human dignity by simply restoring a mutated version, a mutated allele with a wild-type allele in a particular case, I think is probably one that is not in the sights of the drafters of that. The EU Charter of Fundamental Rights has in its sights a particular kind of mischief, the mischief of eugenic practices, in the language of some of the surrounding literature. But it's explicitly focused on states and whether or not that actually would bite in relation to what's so-called liberal eugenics, where an outcome arises from a certain number of individual choices is hard to see as well. Finally, the Council of Europe, Oviedo Convention, the Convention on Human Rights and Biomedicine. Article 13 is the only legally binding international instrument that provides explicitly and prohibitively for the kind of genome modification that we have in mind. And this focuses, unlike the other two, on this notion of integrity of the inheritance of a genetic endowment. In other words, the links that bind the human family together. It's binding law in 29 countries. So does that represent an international consensus? Well, I would say, having been involved with this a little bit and having looked into the origins of the consensus, I think it arose through the deferral of two kinds of question. The first kind of question was whether or not these kind of practices that were in view here could be practiced safely within the current state of scientific knowledge. And I think the answer was clearly, at the time, this is 1997, that they couldn't. And this meant that the second kind of question, about whether they should, really never fell to be discussed. The point is this, that the prohibition in Article 13 of the Oviedo Convention is not a moral consensus. It's simply a prudential one about what science can tell us about the safety of the technique. Insofar as there is a moral consensus, and it's a consensus about the mischief to be avoided, it's not a consensus against the use of all human germline genome editing, but only against that which is aimed at the so-called the selection of persons. Now we're closer to understanding this, to having the scientific knowledge about the possible safety of genome editing procedures. I think it's become urgent that now that we develop the moral knowledge that will allow us to answer that second set of questions. So the notion that there is a consensus against it is, as I've said in previous talks, a canard. How do we govern genome editing responsibly? This is my penultimate slide. There are several things to say here. I'm going to pass over scientific self-regulation because I think it's conceded that the scientists can't do the job. The notion that the market could do it instead, I think, has a number of very, very clear dangers. Markets offer choice, but they don't consider options, which are and very often operate in short-sighted and locked-in sorts of ways. International institutions like the Council of Europe, like UNESCO, have a role to play, particularly at that third level that I described. And the Council of Europe in Bioethics Committee at the end of 2015 made a statement where it said that Article 13 of the Oviedo Convention should be the starting point for an international dialogue. But although they recognized it was the starting point, it was clearly not, in their view, the final word. It's an opening gambit, and they explicitly said a broader kind of dialogue was needed. So, democratic governance, broader societal debate, what does that look like? I just want to mention, this is my last slide, I just want to mention another initiative that you may have heard a little bit about. This was comes out of a meeting in Harvard at the beginning of last year, which tried to give some shape to the recommendation that came out of the National Academies Summit at the end of 2015 for broad societal consensus. I won't say much more about that.
Please, can you conclude?
Because I know I need to finish. All I want to say is that the notion of consensus, I think, is a difficult one, and I sort of come back to Hannah Arendt's problem when you think about forming consensus, and that is what happens when the question of human rights comes up against national sovereignty. And there I think we have problems of international, what I would call geoethics to confront. So that's the meeting that I was talking about. Thank you to the organisers, especially to Francois and his team, and to Hervé and to Luis. And I just finish by saying that the Nuffield report on genome editing in human embryology will be coming out later this summer, so I hope you'll all put your names down for copies of that. Thank you.
Thank you very much.